I have been.
The phone disturbs.
I, that's okay. It doesn't matter. No, no. Give me that PowerPoint. YouTube. Yeah, right. So,
You can say something.
yeah. Can you move it this side, please? Keep it like.
You will be disturbed.
No. Hi, welcome to this live lecture. If you have any questions, feel free to ask me questions. I'll be very happy to answer your questions and queries. Uh, you can type it here and let me know if you have any questions. Yeah. Good evening, good afternoon, everyone. Yeah, so actually, all these things that I discussed in the class, right, whether it is developing a microfluidic device or it is on sensor development and to use it for different applications, this is out of the research experience that I have over last 10 for 12 years. So there is no specific book for case studies. What you can learn from this is if you know the basics of the microengineering and basics of electronics, then can you merge it together to use for different case studies? So as far as I understand, there is no specific book describing this case studies, but other books which can be relevant for this course are already mentioned in the course content. Yeah, Vaishali, that's what I said, that all the list of books are already there in the NPTEL forum. You can just see it when you register for the course. There's a list of books that I have suggested, which you can use for understanding different sensors parameters. But if you're looking for specific things, you please send an email to me and I'll send it back, reply to you with list of books. Generally, we look SMG is one, right? And then we work on different kind of research materials rather than a particular book. So if you talk about research materials, then the best journals that you can refer would be Sensors and Actuators B. That has a lot of information about chemical sensors. You can also look at the Biosensor Bioelectronics Journal, which has also a lot of sensors information. You can then look at IEEE Sensors Journals. You can look at IEEE TBME, which is Transaction of Biomedical Engineering. You can look at IEEE Sensors. You can look at IEEE JMEMS. So JMEMS stands for Journal of Micro Electromechanical Systems. There is a Journal of Micro Machining. So all these journals will help you to understand more about sensors. You have to download the papers. In case you see what happens is not all the universities have the registration to download those papers. So in case if you want one or two papers, I can help you out. And about the notes, as I think somebody is asking about the notes. For the notes, generally after the courses are over, right? Um, the IIT Madras, they ask us to do a kind of voiceover or to put the comments, and whatever I am talking right now, it will come like you can view it on the slides itself. So the course content can be shared only once the course is over. In between, you can only rely on the YouTube videos. That's what from my course at least, right? Yeah, if you want a PDF, I can send a PDF to you, but I have to go through IIT Madras only. Oh, there are a lot of possibilities, Anil, for doing research in the field of microfabrication or clinical research. For example, I'll give you few examples that you can work on. Let's start with the eNose. eNose platform, as all of you know, is used for understanding the breath signature. Right? And from breath signature, What you understand is whether a person is suffering from certain disease. For example, we can use eNOS for diabetes, we can use eNOS for lung cancer, we can use eNOS probably for breast cancer. This is what we are working on. Also, let me give another example. Suppose there is a tracheal inflammation. Tracheal inflammation, trachea means windpipe. Okay, so when the babies are or infants are on the ventilator, the windpipes get inflamed. If windpipes get inflamed, we need to understand in real time whether there is inflammation or not. So you can design a catheter indicator with microflow sensors and haptic feedback to design this particular application. Let me give you another example. There is something called hearing loss. So when a baby is born, the right way to understand is called BERA system, BERA. The system itself is 14 lakh. So can you design electronic module or a head cap that can screen the babies at early stage? Now, why I say this, because only tertiary clinic in India they have this BERA system. Primary clinics does not have it. So can we design a system that can be used for understanding the EEG signal? Right, it's called audio EEG because it depends on audio stimuli. So there are different biopotential that comes out of the EEG. For example, one is AEP, auditory evoked potentials. Second is the P300, which is positive peak at 300 milliseconds, and third is mismatch negativity. So that is another problem. Let's take another problem. So for example, if you want to know the difference from delineate the normal tissues from cancer, can you design a system? So again, there is a different application for that. Let us take another example. If you talk about glioblastoma or meningioma in brain cancer, right? Can you develop a probe that can delineate meningioma and glioblastoma from normal tissues? So that is another example that I was talking about. So there are plenty of research applications that you can work on, Anil, once you understand the basics of microengineering. So let me go to the next question. What is the use of SU8 pillars in sensors exactly? Okay. So the content that we or I was talking about, though at that particular content, SU8 pillars are used as a force transmitting element. That means that, let's say this is a chip. Okay, you can see the chip, right? And if I have if I apply a pressure directly on chip, right? So let's say there is a tissue on the chip. I'll put it here, like, right now. How I make sure that all the sensing element is touching the tissue simultaneously? To make sure this sensing element touches the tissue simultaneously, I'll have SU8 pillars on this. So there is a uniformity, okay? Because tissue is anastrophic. So to have that, I can use SU8 pillars. The SU8 pillar will work as a force transmitting element. That is the role of SU8. Let me go to another example. So Vaishali is asking if needs to send an email. So which email ID we have to use? See, again, if these are very specific questions that you are not able to get answer through the NPTEL forum, then you can send it to my email, right? If the contents or the technical discussion is such that the TS cannot answer, I'll be very happy to answer your question. Vaishali, you can send it to my email. You can find my email through IISc website. I'm sure that by this time you know my name. So you just write down Hardik Pandey IISc, you will get the lab information. Their IISc email ID is also there. Sorry. Yeah. So Vaishali, generally we use IDEs because IDE is easy to fabricate, and what we do is we can use different ways. One is the resistivity measurements, one is the impedance measurement. Now the resistivity measurement will give a overall understanding of the tissue parameters. If you go for impedance, you will understand the further details like cell structure, what are the changes in the cell type, cell morphology, and other things. So from a fabrication point of view, it's easy to fabricate interdigital electrodes. As I have already explained you in the class, right? You take a substrate. Substrate can be glass. Substrate can be oxidized silicon wafer. On this, you deposit a metal, right? Perform lithography and create an interdigital electrodes. There is a difference in the sensitivity when it comes to interdigital electrodes. If the spacing between two electrodes is close, right? Sensitivity would be better. If the width of this electrode is smaller, the sensitivity would increase, right? Now that depends again on what kind of sensitivity you're looking at, right? I'm talking about the sensitivity to understand the change in the tissue parameter. But if you look at the sensor from gas sensor point of view, sensitivity depends on the structure of the semiconducting metal oxide film. That means that if I use a 2D film, thin film, and if I use a nanostructure film, the nanostructure film will have a higher sensitivity, in particular of the interdigital electrodes below it. The reason is very clear that in nanostructure materials, the surface to volume ratio increases, right? So the gas content has a more surface to volume ratio to interact, and that's why the sensitivity would be higher when you talk about nanostructure compared to thin film. When you talk about interdigital electrodes, it's easy to fabricate. So we'll go for IDEs generally. Okay, let me take another example. Anil, an institute which offers research guidance for faculty members who had interest in this field. I'm not too sure what do you mean here, but I think what you are asking is list of institutes which offers research guidance. I think, see, all IITs, all IISc, NITs, they have a lot of QIP programs, particularly in IISc. Because of this pandemic, we are not having QIP. Otherwise, there is a QIP program for 7 days only for engineering faculties or science faculties and for industrialists. So, or engineers working in industries. So that is to improve the quality of the program. So that is that the faculty can come to IISc. They can stay here for 7 days, right? Their travel cost and everything is covered by government, and you will be learning a particular subject for 7 days, 30 hours. So that is how IISc works. There is something called CCE, Center for Continuing Education. You write down CCE IISc, you'll get all the information for faculty who needs guidance in terms of subject. But when it comes to research, I think that is more on your understanding and your connections and your efforts to connect with a particular faculty. Again, there are so many faculties working in so many different research areas. You need to find out which is the research area that you are interested. Suppose you are interested in clinical research, suppose you are interested in microtechnologies, suppose you are interested in sensors and actuators, suppose you are interested in biomedical engineering, right? Or related field, you can always contact me. I can help you to a certain extent. Okay. After that, you have to take it further with your hard work and proper collaboration. So that's what Anil I mean by that. I'm a faculty and okay. Let me see next. Vaishali. Okay, thanks. Okay, great. Thanks, Vaishali. Next is Anil. I am faculty interested in this field. Great. I'm very happy that you are interested. So, what is the thought process like? What you want to do, right? Why don't you send an email? So we can also have a quick Teams meeting, right? So see, depends again. Everyone has their own bandwidth, right? Certain faculty will reply you in few days. Certain faculty will reply in few months. Certain faculty will reply you immediately, right? That depends on the bandwidth and the amount of work that a faculty has. So if you send me an email, I can reply you to your email within 2-3 days of time. Then you can ask me more questions about your interest in this particular field, and I can guide you better. All right, Anil. So feel free to send an email to me. All right. Who else? What are the internship opportunities in this field? So I cannot tell what are the internship opportunities. I think there are a lot of internship positions available within IISc, at IITs. In fact, if you see recent announcement by I think by DST, but I'm not too sure. Just look at the government website. There is an internship programs. So interns would be paid for the time that they spent at in a particular laboratory. Okay. Having said that, how I look at internship is that rather than a summer or winter internship, which is only 2 months, it's very difficult for you to learn a lot of things from my lab. Okay, it requires a bit of intense work. So how I give an opportunity to a student to work in my lab is by giving them a 6 months project internship. That means that they can spend about 6 months. If they are good, right, they can apply as a project assistant, and then if they work for 1 year or 2 years, they can go to a better university, or even apply to IISc for PhD programs and whatever they want to do in their career. The point is, this is how the internship in my lab is defined. But not this is not true for all the laboratories. Some laboratories they give internship for 2 months as well. So it is more like how you approach a certain faculty. Also, it will be great if you can approach the chair of the department. That's the easiest way to know whether there are opportunities available within the department or not. Because see, what I would do is I would see what is my interest and what are the relevant faculties that are working in my field or interest of my field, right? My field's interest. Then I'll make a nice cover letter and send it to those people, okay, of course via email, to see if there is any internship opportunities available within that group. And if yes, then I'll join. So that is how the easiest way to get the internship positions. Vaishali, Sensor Actuators Chemical B listed many papers on IDCs and IDC and forthcoming research over ID, research in sensor development. Yeah, see, the people use different techniques to publish. Okay, we need to make sure that what is the best way to publish, what is the best way to replicate the results, right? It is very important that the repeatability is not put on side. Repeatability, sensitivity and selectivity is equally important. So until you use IDC or use IDE, right, you would not know. But I don't think you can say that IDC is better than IDE or IDE is better than IDC. I think as long as the sensor development is concerned, depending on the research facilities that a lab has, they will go for that particular mode of sensing. So that is what my say is there, or my take is there on IDC's paper. So, but we cannot say that this is an upcoming field. Actually, upcoming field is bioresorbable sensors. Sensors that are advanced sensors. What is advanced sensors? For example, I'll give you an example. Okay, Vaishali. So you have developed a chip, and let's say there is a patient who has suffered from heart attack. Okay. Now, if you know that patient we have to monitor the health of the heart for about 1 month. So it's very difficult to keep the patient on ECG system in hospital. Also, the other system that is available commercially, it is difficult to hold. Can you develop a chip that you can place within the subcutaneous region, just below the subcutaneous region? Subcutaneous region is just below the skin, okay? And the transmitter is outside. This chip will give you a ECG recording for 21 days to 24 days, and after 24 days, this chip will dissolve in the body, so you don't have to reoperate the patient, right? That is the advanced sensor. That is the advanced or upcoming field that you can talk about. Same thing, upcoming field can be a bioresorbable sensor for ECOG monitoring. There is a research field called epilepsy, all right, or stroke. So in epilepsy in another way we call fit, right, where you will see a person is like shivering. That is because of the misfiring of signals in the brain. So if you want to design a sensor that can be placed inside the brain, right, or onto the brain inside the skull, you open the skull, place the chip on the brain, and then measure the real time signals to understand where is the epilepsy episode, right? After a few days, again it dissolves in the brain. That is the advanced field, upcoming field that we can talk about. Okay, so that is what I believe is a forthcoming research rather than two different modalities. So yeah, that's my say again, Vaishali. I don't have much thing to say about that things. Yeah, Sankur, how to measure diabetic with continuously? What do you mean by diabetic with continuously? But I understand what you mean. Let me understand your question. So what you want to know is how can you continuously monitor the glucose level in a diabetic patient, right? So to do that, if you see there are continuous glucose monitoring systems are coming up. It's called GCM, and we have a small project in Birec where we are working. When we say we are working with a medtech company, We are developing a wearable band to measure the continuous glucose from a given diabetic patient. So otherwise there is another thing from um Medtronic, I believe, or something, another company, that they use micro needles to monitor the continuous glucose measurement from the plasma, right? There are ways, but I don't think right now there are any uh, commercial device that can we can use for continuous glucose measurement. Of course, there are few, but there are some limitations to those. For example, there are patch, right? This patch will come off after some time. So there is a difficulty with monitoring continuous glucose from the device point of view. But in a given period, there will be many devices that you will see around you. So, and why do you yeah, that's what I said. To do a continuous glucose monitoring, you have to get HF clearance to find out a number of subjects who are ready to give a consent that every 1 hour I'll try to pick your blood, right, and measure the glucose content. Very difficult. Also, there should be device that can correlate with that. Also, there should be gold standard that you have to correlate with our device. So there are a lot of limitations that we need to overcome to bring certain kind of device to market. So that's the result. Okay. Um, okay. Thomas Mohan. Currently, do you guide research works in invasive tests only or non-invasive also? See, I have two postdoctoral research scholars. One, she finished her PhD from IIT Ropar. Another student who finished his PhD from IIT Madras. And I have about nine research students, which is PhD students, whom I am guiding right now. And they work from in a broader umbrella. They work on healthcare technologies, where they work on non-invasive methods. They work on invasive methods, not really invasive, but minimally invasive methods. And in one case, it is invasive methods as well. So I work on a broader domain from non-invasive to minimally invasive, and to certain extent to invasive as well. So that is what I guide. And of course, there are few MTech students, about four, and about eight project assistants. So there's a lab of 24. It's a huge lab. Yeah, okay. Vaishali is asking, what is the role of MEMS sensors in the field of automation? How can we see guidance systems? Okay. So MEMS are used in robotics and automations. Okay, so when you say automation, automation also can be linked with robotics. If you see a lot of robots, right, that are upcoming, they don't have any flexible sensors similar to our hand, right? For example, our hand has a lot of force sensor as well as haptic. That means that if I pick this mouse, I know how much force to apply, and I can feel the texture of this whole mouse, right? But can a robot do the same thing? Right? So can you develop a MEMS-based flexible array of sensors that can give a real-time force feedback, and also can you convert it to haptic feedback? That means let's how much amount of force a robot is applying, can a robot feel it? Right? It's very difficult, no? Because when you apply a force, you feel it, right? So haptics is very important work that is ongoing research field. Automations, you have used MEMS transducers that are used in automation. MEMS actuators are used in automation. To an extent, SMAs are used in a catheter for maneuvering the catheters. SMA stands for smart actuators, and you can use it for robotics again, for maneuvering the catheters like tubes, and so on and so forth. So yes, that is as far as the automation is concerned. Yes, MEMS are used significantly into automation. I'm just giving a few examples, that there are many. There are pressure sensors that are used in automation field. There are force sensors that are used, and then there are microfluidic devices that are used to a certain extent to understand the to understand the change in the rate of the fluid. A lot of other things are used. So MEMS has a significant role. Again, if when you see the just your modern day automobile, right? You open it and you will find so many MEMS transducers and gyroscope and pressure sensors and so on and so forth. So MEMS has a huge role in automation. Now, how can we seek guidance in this domain? I think again that depends on the faculty's interest of to work in that particular domain. So I don't. I of course work on catheters and a bit of biomedical robotics, but there are a lot of faculty in the institute who extensively work on automation and product design. So you can find it out either in mechanical engineering or in CPDM, right, or applied physics, instrument and applied physics. So we'll identify what kind of faculty's interest is there on using MEMS for automation. So that's how we can find out guide. Yeah, yeah, great. Thanks, Thomas. But if there are certain specific questions, like you see, which are not related to this particular topic, right, the course, right, uh, then you can make a list of questions and send me an email. I'll try to reply to your email as soon as I can. Okay. So Ankur is asking that I am doing okay. First of all, you can call me Hardik. Okay, don't call me sir. Um, uh, I I am doing MTech in electronic systems and communication. After that, yes, yes, definitely. See, I have this time I have hired a student for PhD who has a MTech in VLSI, right? Uh, and we have people who has MTech in control systems and instrumentation, right? Who has MTech in that, and then now they are doing PhD in biomedical, so or healthcare technologies or MEMS. So there is a definitive chance of getting a PhD admission in this domain, provided your basics are correct. Correct. What generally we look when we interview a student is how good a basic of a student. So basics are like we even ask you basic math in the interviews. Okay. So it is it is nothing to do with what is your background. Okay, EC electronics in general, right, can be several things: electronic instrumentation, electronics communication, right, electrical electronics engineering, instrumentation engineering, right, applied physics. So in fact I have hired students from BTech in mechanical engineering for pursuing direct PhD if they clear CARE exam. So Ankur, you have a sure such chance of getting into PhD positions in most of the universities, provided you do well in your interview. Yeah. So if you don't have any questions, then I can stop streaming. But if you have any, please feel free to ask me. Okay. Okay, that's my email ID. Uh, any open source software for MEMS? Not really. Uh, see, software, open source software, I don't know, but if we can use COMSOL as one of the software that we extensively use in our lab, right? But there is a student version, and if you are working in a institute, then you can buy a license version. It's not too costly. It's about 1.5 to 2 lakh per software. So you can try to get that if your institute provides that one. Yeah. Yeah. So there is no beginner or expert in understanding software. Yeah, that's what I said. For when he said MEMS, I told you that I generally like to use console, uh, and uh, there are several modules of console, right? Which one can use. You have to select the basic modules for students. There's a student version actually. So if you are interested, I can send you the link to get that particular modules, right? Uh, so that it will be useful for you if you like to do that. Uh, Let me just find it out another way. Mm, to help you out, if I can connect with my student, but just give me a second. So the reason that I'm talking about right now about console is that we use it extensively, okay, and then there are other software as well like Coventovware and other softwares, but I generally prefer console over multiple things because there is a lot of physics involved in deriving to that particular module. So your understanding about the or using the physics for solving a problem is very important. It will help you to understand the equations. It will help you to understand why certain things are changing when you apply a pressure. For example, if you take a pressure sensor as an example, right? So I believe that that is how COMSOL is better compared to other things. But this is only based on my experience. Okay. So if you want a specific module and what is the cost, I can get it because I don't have it in right now. I don't have that values in mind. So I can let you know specific modules uh, and the cost associated with those modules once I cross check with the company. So send me an email. In fact, if you can share your email, Anil and Vaishali, with each other, you can send me one email with copy to, if Vaishali is sending me an email, copy Anil on that. If Anil is sending email, copy Vaishali on that. And ask me the same question. I'll give you a detailed reply because I don't want to give you some wrong numbers when it comes to buying of modules. There may be a possibility that you can get a free version for 14 days, right? So I can connect you with the right people in that company as well. No, no, no. You can do simulation of the yeah. Of course, you don't have to do fabrication, but fabrication is different, right? Fabrication you have to come here and get your hands dirty. Fabrication no need of looking at software and doing fabrication. I never did that in my life, so I would not suggest you to look at some software and fabricate a device. Reshmi. COMSOL can be used to put layers on each other, right? For example, pressure sensor, right? You have to give details to that, so to create a structure, and then you can understand whether the structure for that given layer will work or not. Now, the fabrication process for that depends on your understanding about the subject. But I don't think, as long as I am concerned, I don't use any fabrication software per se. That's the only way to learn fabrication is by attending the clean room, getting trained on different modules, right? From e-beam operation, to thermal operation, to lithography, to DRI, to RI, to PECVD, to LPCVD, wafer bonding, wire bonding, right? And then characterization like SEM, XRD, TEM, and so forth. So until you until you have access to clean room, it will be difficult for you to really understand the process of a fabrication. So you are free to ask me a few questions, please. Yeah. Okay. So yeah. So see, I prefer console. Doesn't mean that you have to work on console. Okay. You want to use Conventor where you can do simulation of application. My point is, uh, there is no point of performing or using a software for fabrication, because it will not give you the understanding of how the device is fabricated. There is no way, believe me, there is no way until you attend the experimental labs, you go to the clean room facilities, you learn the systems. Take a silicon wafer, holding in hand of silicon wafer is very different than drawing a circle on the software. Okay, so that's what I meant by not liking of, or I don't like a fabrication software, and I like a student to go to the clean room and learn the thing. So that's what I meant, Reshmi. Suggest a book for micro and nano fabrication. There are many from Springer. There is one from micro and nano fabrication by Yan, that you can look at, right? There is another one by Getsen, micro and nano fabrication, again from the Springer. So look at different books. More or less, they will teach you the same thing. Okay. Yeah. So yes, Vaishali, there will be difference in the simulation results and the experimental results. There is you should perform simulation with utmost care, but when it comes to fabrication, the process and the recipe is very important. For example, like what Reshmi said, right, that she can do a fabrication using a software, right? Yeah, I'll get back to you, Reshmi, your question. So I'm giving example of what Reshmi said, that okay, we will do a fabrication study of a particular sensor, right? Then what happens is that she will select, let's say, silicon wafer 4 inch, diaphragm thickness to 150 microns, right? Sensing layer 10 nanometers, right? Now, or let's say 1 micron, and then when you actually fabricate it, the diaphragm thickness would not be 150; it comes to 145 because of the recipes that you are using, right? Because of the process is not correct. So most of the time, if the simulation results that are given to you will be correct, if you know the simulation, the fabrication results will not match simulation because of the recipes that are involved in the fabrication. Either there is a you are not putting your clock correctly. That means that if I want to etch a silicon wafer for 1 hour, I'll etch it for 1 hour 2 minutes, and in 2 minutes there's a difference of diaphragm. This results will totally change with simulation. All right. But what you can do is, now you have a fabrication, you have done fabrication, okay, and you got 150 microns instead of that you got 130 microns. Then you can change the value in simulation, see whether for 130 microns the same values are coming or not. That's the only way that you can do. But as long as these lines, that is like the system and the processes are meant for only a particular sensor, the repeatability is very difficult. That's why the entire line should be dedicated only for fabricating one single sensor. Or if you see the fab labs, the entire lines are fabricated only to design the transistors, right? Design the electronic circuits, right? So they don't do multiple things using the same line to avoid any mismatch in recipes. Okay, so that's what I also found, Vaishali. That's a very good observation, that when you do simulation and when you do fabrication, there is a difference in the results, right? So, okay, Reshmi, actually in our college we are using COMSOL, but publication in Sky journals is very critical. I don't agree with you, Reshmi. As long as you have a licensed version. If you don't have a licensed version, you should not publish. It is as simple as that. I have published papers with COMSOL and I have a licensed version. Student has a licensed version, so we are using it, right? So yeah, with a licensed version, you can publish in good journals. We only publish in top journals, at least Q1 journals. So for guys who don't know, there are four quartiles in a journal: Q1, Q2, Q3, Q4. You should start and focus only on Q1 journals that are good quality journals. Okay, certain times Q1 and Q2 overlap, so it's fine. Never publish in Q3 or Q4 journals. So I kind of disagree with you, Reshmi, that COMSOL simulations cannot be published in a Q1 journal. Of course, what we do is we do the model, we do the simulation, we do the fabrication, we do experimental testing, and then we publish. So it is accepted. I have published in IEEE TBME, I have published in Biosensor Biotronics, as long as I remember, and also to an extent to IEEE GMM. So we are using it. I am little bit surprised why you think that you cannot use it. Okay. But yeah, again, let me reiterate. You have to use a licensed version. If your college is using a software, it is better that you connect with a company and get a licensed version for the entire college, right? Just get one licensed version on which a certain student is working on. That should be enough for you to qualify to to publish papers using the simulation that you get from the licensed version of the software. Of course. Oh, sorry. So I didn't understand, Reshmi, your complete question, but I'll come back to Vaishali. So Reshmi, yeah. So we have assumption, but in science, yeah, of course. Yeah. So only simulation results would not be enough for you to publish a paper. You have to fabricate devices. So it's it is mandatory. I'm sorry, but that is how it is. Because what happens is that if you bring a very novel kind of work which nobody has tried, and if you show by simulation that this can be useful, they will consider. If your simulation has lot of mathematical model backing up your simulation, then you have a chances of publishing. But most of the times, what happens is if you simulate a sensor, right? And you want to publish it, they will say fabricate a sensor. So fabrication, when you do fabrication, when you do testing, iteration, these results are very important to publish in Q1 journals. So yes, that is very important. So I really cannot help uh, through this particular mode of helping you out, but there is a way to collaborate. If you have a certain funding, you can use it for fabrication for that particular sensor or device or transducer that you are looking at. Okay, let me go back to Vaishali's question and then I'll take Anil's question. So, how much variance we can consider for virtual and actual results in that? Okay, so generally, my guide used to teach me is that Hardik, if you do simulation with 100% accuracy, your experimental results will come to 75. So that's what was the variance in his laboratory, right? With the system and the facilities that we have established in IISc, and to a certain extent, I use the NanoFab lab, which is our center facility. Um, we reach close to 10%. Okay, but still there is a variance around 10%. Okay. So Anil, what about ethical clearance for testing on humans? Yeah, what about it? I Oh, do you have any tie-ups with hospitals? Oh, yeah. Yeah. Sure. So yes, for you to use a device on humans, you should have a TR clearance, one. But depends on what kind of device you are proposing. For example, if I want to understand the tissue properties, right? Let's say we are talking about prostate cancer, right? So if you want to understand the tissue properties humans, then if the tissue is out of body, then I can test it in my lab, provided that I have a clearance to use those tissues. Now, who will do that operation and who will give us the tissue? It will be doctors. So I should have a collaboration with doctors. They should apply for ethical clearance in their hospitals, and then that ethical clearance I have to again submit it here in IISc. It goes through the Institute Human Ethical Clearance Committee, and they just look at, okay, you have ethical clearance, so we also approve, okay, more or less. And then there should be biosafety approval. That means that my laboratory is safe enough to use those tissues to perform experiments. So this is a process for human samples. When you implant or put a device inside a human body, before that you have to show that it is working well with animal model. So you have to get animal ethical clearance, which is called IAEC, Institute Animal Ethical Clearance. Now depends on what kind of animal you are proposing, right? Suppose I want to use the pig's heart. First I want to understand how the tissue properties of a heart is concerned. As long as it is ex vivo, it is outside the body, ethical clearance becomes easy. If it is inside the body, in vivo, ethical clearance becomes difficult. But yes, you have to have collaboration with hospitals. I have about 21 surgeons with whom I collaborate as of now, and we have ethical clearance for using the human breast tissues. We have ethical clearance for using pig's heart. We have ethical clearance to implant the chip in rat's brain. We have ethical clearance to use sheep trachea to test the efficiency of our catheter. So that's what you have to collaborate with a doctor, clinician, if you want to work on healthcare and you want to get the ethical level. Yeah. So let me give an example, Vaishali. See, let's take an example, ultrasound transducer, right? So as the as the frequency increases, higher the frequency, smaller the transducer will become. Okay. Same way, wavelength has also a relation with the sensor dimension. How much is the optical window of your sensor is very important. So and of course, where it comes more towards the optics, so you can see a different kind of LEDs which falls in NIR region, 850 nanometer, 930 nanometer, 1050 nanometer, and when you look at the specification chart, you will understand the dimensions and the variation that they use to create the change in the wavelength. But there is another way to look at different wavelengths is by using filter. All right, you have a LED of a, say, of certain wavelength, you put different filters and you will have a different wavelength. So there are ways, but yes, there is a relation. You can see this kind of I believe this questions that you're asking is good. I'm very happy that you guys are asking questions, but at a of course, some of the things you may not know, but other things is very easy to even identify or get it from Google. All right, very easy. A lot of relationships, equations, different softwares that are available, which one we have to use it, which one gives better results. Yes, something you will not know is how to fabricate a device, whether I require fabrication along with a simulation to come up with a result. How this is a good question, that how accurate my device parameters would be compared to simulation. These are some interesting questions. But again, not demeaning you, just pointing is that you can find out a lot of answers to your questions even using Google. Okay. All right. So I believe that you don't have any more questions. I think actually there are few students who only four actually, Anil and Vaishali, Reshmi, right, that asked questions, and I think in between Anil, Ankur asked a question, Thomas asked a question. So yeah, and then I don't know the name, but technical knowledge in electronics. So yeah, referred many papers from sensors how to with the sensor dimension. Yeah, see, if it is a very specific technical question, Vaishali, I'll really request you to send me an email and I'll help you out, okay? The specific question related to fabricating sensors, how to select the dimensions, and what is the application that you are looking at, right? What is the final application? Are you looking at the wavelength that you want to select for particular application? Various papers giving different dimensions. Is there a way to? Yeah. That's what I'm saying. That please send me the queries with details like okay, Hardik, I want to use this particular sensor for understanding tissue properties, right? Now I am creating this sensor, how the relations between dimensions and wavelength would be. Can you give me a few examples of those sensors? Can you show me how to fabricate devices? Be specific in your question, but ask me a particular set of questions so that I can help you out. This is too difficult for me to help you out in another 5 minutes left. So I would really suggest you to send me an email. I will definitely reply to your email, Vaishali. I have already sent the email, shared the email with all of you guys. Show me the papers also. Whatever papers you are referring, attach in the email. send it to me. I'll get back to you as soon as I can. Generally, I'm very fast. You don't have to remind me. In 2 days, 3 days time, I'll get back to you. Because optical sensors are different than chemical sensors. Optical sensors are different than the transducers, right? And again, in optics, what kind of sensors you want to use and what is the application. That is very important to start with. That what kind of wavelength I am selecting, why I am selecting that wavelength, right? How it is relevant to my application. So all these things we can only understand when we discuss in detail, or I have a detailed understanding of what exactly you want to work on. Once you give me those details, I'll get back to you, Vaishali. No problem at all. Yeah, thanks, Anil. Okay, so I think we will stop it here. It's already close to 5, and I don't see any questions, right? So enjoy the remaining course, all of you guys. And like I said, send me an email, Vaishali, Anil, Reshmi, if you have any questions, Thomas, um, and I'll reply back to you via email. Yeah. So till then, you all take care, right? Have a nice remaining course. Do well in your coursework, right? Uh, don't die every day to live, right? Be happy and enjoy the remaining part of the course. I'll catch up with you through NPTEL forum if required, or via email if required. All right. Till then, you take care. Have a nice